While we're waiting for people to come in, are there any issues that anybody needs to ask? I'll make some announcements, but I'll wait until. Well, I guess it's time enough. Okay, a few odds and ends things that I want to, announcements that I will probably, some of which I've already made and some of which I probably will make by email, but I just want to do it anyway to be sure. Okay, first of all, assuming that I finish today's lecture by the end, we will have a review session on class time on Friday instead of a lecture. By review, I mean a question and answer session. You have to come with questions that are, usually what happens is there are no questions at the beginning and then people start thinking of them. But anyway, that's what that will be. If I don't finish, then I'll discuss with you at the end of the hour what we'll do. Secondly, just to remind you that the neuron vision study questions are due at midnight tonight. Also, I sent you an email about this. I'd like you to do teacher ratings for this first half of the class by Saturday midnight. You get 3 points quiz credit for doing that. So please do it. You can't, as I explained in the email, I can't tell who responded how. I just can tell whether you responded. But I will get the average, total numbers of points people, the answers to the questions, but not tied to individuals. It doesn't give you an opportunity to make any free-form questions. You can do that at the end of the class in the official thing, but this, I want to do this instead. Midterm is Monday. I think you all know that. Class time here. Scantron forms will be provided. Bring number 2, more or less number 2 pencils and erasers, which reminds me. Get some extras from the storeroom so somebody breaks their, and can I do erasers. There will be only 2 questions on learning on the midterm. Most of those questions will be on the final. There are exactly 2 questions. And the study questions on learning will be posted after the midterm. I don't want you distracting yourself with them now. The questions that will be asked will not be difficult questions you will need to study things for. Okay, and I just asked you a minute ago, any other issues? Anything else? Yes? How many questions? About 45, roughly. Yeah, it's about 45. It's all multiple choice. Yeah. And it's there's no. I might forget to say this on the exam, although I'll try to remember. It's if you don't know the answer, guess, because there's no penalty for guessing. Don't ever leave a question blank. That's you always have a chance. Okay, so we were talking last time about mechanisms of learning and mechanisms of making learning long-lasting. And we pointed out that protein synthesis is not needed for learning, but is needed for making the learning long-lasting. We then started delving into the question of what the cellular events are that drive or initiate the protein synthesis that is needed for stabilizing memory. That's what we were in the middle of doing last time. We had put this picture back, which I had dragged up from the little mini lecture that I put on YouTube at the beginning of the quarter on genetics. For our present purposes, the important point is that this sequence leading to short-term transient transcription commonly depends upon some sort of stimulus to the cell. It might be synaptic input, which in turn causes the production of a signaling molecule, which in turn does other stuff. The first of these signaling molecules to be discovered, so-called second messengers, the first messenger being the input to the cell itself, the so-called second messenger, the first of the so-called second messengers discovered was this stuff called cyclic AMP, CAMP. That was the first one that people looked at in the context of learning. And what I told you last time, and that's more or less where we stopped, is that cyclic AMP did turn out to be crucial for getting long-term learning. And one experiment, which one piece of evidence for that was this experiment, which was not on learning itself, but on long-term potentiation, in which, this just reminds me of the situation, some external stimulus to the cell, in this case it's synaptic input, causes an increase in cyclic AMP, and in various kinds of cells, cyclic AMP can do various things. In a liver cell it does one thing, and in skin cells it does something else, and in some brain cells it does one thing, and in other brain cells it does other things, but the ones that are involved in learning and memory, it's involved in, at least in part, in stabilizing memories, as well as doing some other things related to learning. In any case, in this experiment, what was done is LTP was induced here, with red bar pairing the pre and the postsynaptic activity to cause the Heb synapses to get stronger onto the cell and to cause a stronger synapse, a more effective synapse. And in a normal animal, we see several examples of this already. I'm not sure again whether this is the same experiment as I showed you last time or not. I can't remember, and I don't remember whether this is amygdala or hippocampus. But in any case, after this pairing procedure indicated by the black arrows, we got an increase in the strength of the synapse and the effectiveness of the synapse, excuse me, which decayed some but then stabilized, lasted a long time. If exactly the same experiment was done, but having injected something that prevents the action of cyclic AMP in the cell, then you again get the LTP okay, but it falls away over a period of an hour or so. And so you don't get the long-term learning. And there are lots of other experiments that lead to the same conclusion, that cyclic AMP is important, at least for some kinds of learning and some kinds of cells, for stabilizing memories and stabilizing the synaptic changes that are responsible for the memory. So, okay, we're interested in this process of stabilizing memory and this process of getting the protein synthesis that is responsible for stabilizing the memory going. So the first step is, got something to do with cyclic AMP, but the cyclic AMP doesn't do anything direct. Its action is always indirect. And in particular, its action is to cause the tagging of these transcription factors or regulatory molecules, transcription regulatory molecules, which then, once they're tagged, bind to a particular spot on the DNA, which, once that happens, causes downstream genes, immediately downstream genes from that spot to be transcribed. And then subsequently, that makes messenger RNA, and that also has to be ultimately translated in order to make protein. So in this case, what we would like to do is we'd like to get hold of this guy here, this transcription factor. Well, nobody, as I told you last time, nobody knew where it was or what it was. But that was the next logical step, at least the investigators who worked on this thought it was the next logical step in unraveling this whole thing. And so they found it in a very indirect way, and that's the shaggiest part of the shaggy dog tale. The logic went something like this. This tagged regulatory factor, this tagged transcription factor, has to bind to the DNA to get the transcription going. Well, there is some sequence of bases or nucleotides on the DNA that tagged molecule gloms onto it. It's a specific sequence. That sequence was also not known, but they gave it a name. They called it the cyclic AMP responsive element. So that was the name they gave this presumed DNA sequence, and they didn't know what it was. So CRE, cyclic AMP responsive element. Then, the next step in the story has got absolutely nothing to do with the study of learning. It turns out that cyclic AMP is used, as I said a few minutes ago, is used in lots of different kinds of cells for studying lots of different kinds of transcription going. And and a lot of these cells had been studied over some period. A lot of these kinds of proteins that are made by these various cells have been studied over a long period of time. And here's a list of some of the kinds of proteins whose gene transcription is stimulated by cyclic AMP. And these, somatostatin, parathyroid hormone, protocephalin, you don't care what these things are, but they're things. If one looked, and one knows where the genes for these things all are. One doesn't know where the genes having to do with learning are, but one does know where one did know where the genes for making these proteins were. It turns out that if you decoded the DNA and looked just in front of each of these of genes for each of these guys, these are all independent genes. Look just in front of each of these genes, and looked at the sequence of bases in the DNA, you discovered that all of them had a little short sequence of 10 or so bases that were all very similar to one another, so-called consensus sequence. And so the guess was that was the sequence to which the tagged regulatory protein here, which is purple in this slide, was binding. And it turns out this is just a short sequence of bases, and chemists can make that stuff. So they made it. They make a whole pot of it. So they make a pot of this fake CRE. So this is not in DNA. It's not going to do anything. But this is the sequence in DNA to which the tagged regulatory protein that we're looking for binds. So they made a bunch of this stuff. Now, once, as I said here, once the sequence is known, you can make it chemically if it's not too long, and this isn't too long, not too long in sequence. Okay, once they had this stuff, then what they did is they wanted to use, since the molecule they're looking for will stick to it, they wanted to use it as a lure to fish for the protein they're looking for. So what they did is they ground up tissue that was expected to have, sorry, let me go back a minute. Some terminology. So the sequence is called the cyclic AMP responsive element to which the thing binds, or CRE. The protein itself, which when tagged binds to the CRE, was named the cyclic AMP responsive element binding protein. Again, they have no idea what it is, but they're just giving it a name so they can talk about it. And it's abbreviated because that's a long mouthful. They call it CRED, which is a term you do have to remember because we'll be talking a lot about it. So what they wanted to find is this CRED molecule. And somewhere there's a gene that actually makes this molecule, and it's hanging around in the cell normally. Again, if you stimulate the cyclic AMP, it gets tagged. Once it's tagged, it gloms onto the CRE sequence, and that sets the transcription of whatever downstream genes there are in motion. Okay, so we're looking for that protein. We're looking for the CRED. The cyclic AMP responsive element binding protein. So what they did is, they ground up tissue expected to have the CRED molecules. This would be any tissue in which cyclic AMP stimulated transcription occurs. And it was not brain. Well, it may have been brain, but it was brain, nothing to do with learning. It was various tissues that they knew would have this. And we're going to indicate those molecules we're looking for, those CREB molecules, by purple in this slide. And then there'll be other proteins, of course, too, and I'm going to indicate all the other kinds of proteins as an open circle in this slide. And you grind it up, and then you mix it up in a test tube, or some sort of reaction vessel, with this synthesized CRE that we've just talked about making, these artificial CRE sequences, the cyclic AMP responsive element sequences, DNA sequences, which I'm going to indicate here by a little red line. Together with some cyclic AMP, which I'm going to indicate by a green square, together with the tag. which I'm going to indicate by a light blue triangle. This tag is normally phosphate, but that's beside the point for our purposes. So we smush that stuff all up together. And what we expect to happen now in this test tube is that since all the ingredients are there, the tag will glom on, sorry, the cyclic MP is, sorry, the cyclic MP? No, there it is. Cyclic AMP will cause the tag to glom on to the Krebs molecules, which we don't know what they are, we're looking for them. And they will stick to these guys, and there's one other trick we do. These CRE sequences that we synthesize, we also make radioactive, so we can spot them. That's easy for chemists to do. So all these little red guys, to which we hope or expect the Krebs to bind, are all radioactive. So now we have in this test tube a bunch of molecules, but among the things that are there are some radioactive CRE sequences bound to some Krebs, some tagged Krebs. Okay, now having done that for a little while, we have these molecules. We now take the stuff out and put it on a piece of basically blotter paper and spin it in a centrifuge or do other procedures which will cause the various proteins in the smush to separate according to either molecular size or molecular weight or some characteristic like that, and some other physical characteristics. So we now have a piece of paper, blotting paper, or sometimes some jello, where you've got blobs of protein. But one of those blobs of protein is the one that has the exact molecular weight and size associated with this glomp here. And those are the guys we're looking for. So we now have isolated from everything else a little pile of molecules which has basically our artificial CRE sequences, which we made, our radioactive CRE sequences, and that's how we can spot them because they're radioactive. Glommed onto this molecule we're looking for, the purple guy, the Krebs, and the tag. Once you've got that, you can dissociate the stuff so that you have these things separated again, and you've got a pot of the Krebs molecules. Once you've got the Krebs molecules, this is a protein. Easy to sequence proteins. That was known they knew how to do that long before they could deal with DNA. So they can sequence it, find out the amino acid sequence. Knowing the genetic code, they can read back and find out what the DNA sequence is, the code for it. Once you know the DNA sequence, you can find it on the gene. Once you can find it on the gene, you can breed, with using molecular biology techniques, you can breed a mouse that has it in a defective form so the gene won't work. That's called a knockout mouse. Or you can breed a special mouse that has it in some altered special form called a transgenic mouse. But we're interested in knockout mouses here. So we make a knockout mouse, or we didn't, these people did, made a knockout mouse that couldn't make Krebs. Okay, and what we're predicting is that guy's going to have lousy long-term memory. Perfectly okay short-term memory, but lousy long-term memory. Okay, that's the next part of the story. So next we're going to show you that, in fact, it turns out to be right. Krebs is, in fact, needed for long-term synaptic change and long-term memory. This is what I just said to you a minute ago. Having found the protein Krebs, one can read back the genetic code for it and thereby find the gene. Then with about a year's work and sufficient dollars, this has gotten cheaper and cheaper. It used to cost about $1 million. It doesn't cost that much anymore. One can make a mouse that has a non-functional form of the gene, a knockout mouse, and so that was done for Krebs.
Why is it cheaper now?
Different techniques are used.
Okay. It's just breeding mice?
Well, no, it's not. It's more than just breeding the mice.
No.
It says right there, you may have noticed, if there's time, tell you the story of how you make a transgenic mouse. There's not time. That itself would probably take a whole lecture, and I wrote at one point, and I never had a chance to get it, so it's like checking out slides out. Okay, so here's the experiment that was done on these Krebs knockouts. And in fact, it was done by it was not actually done here at UCLA, but it was done by a fellow named Alcino Silva's lab, who is now at UCLA and who still does this kind of work. It was a very famous experiment. So we've got these Krebs knockout guys, and they're tone fear conditioning. That's why part of the reason we're talking about one of the reasons we're talking about tone fear conditioning, or about fear conditioning. So they're classically conditioned to be afraid of a tone. When you present the tone, a normal mouse will freeze, and they'll do that both right after they're trained and for days and days and days and days after they're trained. And a normal mouse, here shows a normal mouse who was conditioned The tone was played, and we looked at him 30 minutes after he was conditioned. So that's still pretty short time after conditioning. And this is how much freezing occurs when you present the tone. So the animal's sitting there, you present the tone, he freezes. The normal mouse freezes, so this is just 6 minutes. We leave the tone on for about 6 minutes. And the Krebs knockout, the low Krebs mouse, also freezes. So that's fine. But that's what we expect. We don't expect the Krebs to be involved in the learning. We only expect it to be involved in long-term memory. We make the same measurements now, even 2 hours later, as short as 2 hours later, or 24 hours later. Then what happens? The normal guy still looks the same as he did at 30 minutes. But the guy who the Krebs knockout doesn't. It's not a total knockout, but it's a very substantial knockout. And so he's mostly seems to have forgotten to be afraid of the tone. So he's not very afraid of the tone anymore. So that's exactly what we predicted. Here's a similar experiment, terrible experiment actually, done with LT on long-term potentiation in the same population of animals by Sola's lab, in which they got LTP, and then in the Krebs knockout they get LTP again, but it only lasts for less than a couple of hours. So that's all hanging together very well. So this Krebs stuff seems to be, let's see, what do I have next? This Krebs stuff seems to be doing what we thought. It appears to be the transcription factor, or the evidence is consistent with the idea that it is the transcription factor that's setting the protein synthesis going that is needed in order to stabilize long-term memory. So that's quite an accomplishment, but it goes a lot further than that, yes.
What are the proteins that activate the transcription?
That's a very good question, and there's not very much known about that. That's what you think they would have gone after first. And there is some work on that, but there's no neat story to tell. They are things like synaptic components of synapses and things like that. There's molecules that are going to synapses. That's known, and there are some things known, but there's no neat story to tell.
Do they know, like, when we recall a memory, what's happening, like, at all?
Well, it depends it's going to be different for different memories. For example, it's at least going to be different for memories having to do with declarative versus explicit versus implicit memory. In the case of this kind of memory, with a classical conditioning memory, what is happening is that the stimulus that you present that is going to cause, that is potentially going to cause the conditioned response, stimulus gets represented in the brain, and that fires some neurons which hook up with the neurons that cause the response. And it's those synapses getting strengthened that is the memory, and because they're stronger, you make the response to that stimulus that before did not cause anything to happen.
Recalling, like, someone's face.
That's a different, okay, now we're talking about declarative memory. That's less well worked out. That information is probably coded initially in the hippocampus, and there are ideas about it, but it takes too far afield. There are ideas about it. It's not nearly as well understood as this time, which is the reason I'm talking about this time. Okay, so we have this Krebs stuff. Now, having discovered Krebs, people were doing this mostly just because they wanted to work it out. These are pure scientists for the most part. They just want to understand how it works. In the back of their minds, they have the idea if they could really understand long-term memory and how it's produced, that it might have some practical value. But that's not why most of them are doing it. But we'll see that story changed pretty fast. You'll see in just a minute. But this is just being done for not for practical or so-called translational reasons. But in the process of studying this Krebs, they discovered there were genes that made Krebs, but there were also closely related, very closely related molecule, also called Krebs, that was a little bit different. It did a different thing. In fact, it did exactly the opposite. Whereas the Krebs that they had first found caused transcription to start, the Krebs they then found would prevent that from happening. So these things have been called various things. I'll call them Krebs 1 and Krebs 2, or more often for our purposes, I will call them, although this is not said this way in literature, I'll call it good Krebs and bad Krebs. Okay? So good Krebs was discovered first, and then it was discovered there was also a bad Krebs that would prevent the action of good Krebs. Well, that's interesting. Once that was discovered, that led people to some ideas about why it is that space practice, space learning is better than math, which is where I'm directing these lectures. So let's go back to that for a minute. First of all, the discovery that space practice is better than math practice occurred a couple hundred years ago now, I guess it is, by a guy named Hermann Ebbinghaus, a German fellow. And he's actually the first experimental psychologist. And he did all his experiments on himself. He invented the so-called nonsense syllable. And then he would make lists of nonsense syllables, which he would try to memorize. And then he would see if he could remember them. He basically would memorize them. He had the list. He had a piece of paper. He would say, let's try to remember all the nonsense syllables on his list and the variants of that. And what he discovered is if he practiced, a lot over a short period of time, he could learn it just as fast or even faster than if he just gave a little bit of practice every day. But if he gave a little bit of practice every day, he remembered it for a long time. And if he gave mass practice, he didn't remember it for more than a day or so. So mass practice yields little long-term memory. Space practice is much better, is what Ebbinghaus discovered. When I was preparing these lectures, I asked my wife, who happened to be sitting at her computer next to me when I was preparing this lecture, several years ago, see if she could find me a picture of Hermann Ebbinghaus. So she came up with that. And I looked at it and said, I don't think his name was Jurgen. I said, I don't think that's the right guy. So she looked around some more, and she found the real Ebbinghaus, Hermann. So the one on the left is Hermann. But I submit that there might be the same family. Anyway, who knows?
Professor,
Yeah.
that's kind of like a weak basis for such a strong conclusion. But like,
Which?
Yeah, that one guy did self-experiments. That that seems like.
Oh, he did lots of experiments in lots of ways. He didn't do it once. He did this for years and years and years.
The fact that they're on himself.
It could be that he was the only person for this law was true. That is absolutely correct.
Or even that like he already had an opinion about how.
He didn't, though. Well, you could, okay. He either did remember or he didn't remember it. You have to argue he cheated in doing his own experiments. And why the hell would he do that? I guess he could. I mean, why even bother doing experiments? Why not just publish lies if he wanted to promulgate an idea that he had ahead of time? All of that is possible. But I submit that the more less cynical, more plausible interpretation is that he actually got it right. I mean, it turns out he did have it right. And that's been shown thousands and thousands of times since then, and you all kind of know it yourselves from personal experience. Unless you have photographic memories. Some of you may be a little different. That's the point we will actually come to in a minute. Kind of. Okay, so we got good cred, bad cred. We have the spacing massing thing. And at this point, there was a guy, there's been lots of work done on learning, not only in mammals, but people like to use lower animals for these kinds of experiments, not because, well, for a variety of reasons. But one of the reasons is they know, or at least think they know, that the nervous systems of lower animals are simple. And they think it might be possible to figure out things on them that they can't figure out on higher animals. Or sometimes there are some kinds of usually lower animals that have very special features to their life cycle or biology or brains or whatever that make it particularly easy to study some particular thing. And that was the case here. People that were interested in studying learning wanted an animal, to study an animal that could learn, but that they could easily do genetic experiments on, because they wanted to see what genes might be involved in learning. And so the obvious animal to use, you can use mice, that's kind of what people do now to a considerable extent, but a much better animal to use is the geneticist's pet, Drosophila, the fruit fly, which basically breeds really fast, as you all know if you've ever left any rotten fruit in your kitchen. Actually, I did that over Thanksgiving, and we've only just now gotten rid of the fruit fly population in our house. In any case, so people wanted to see if they could figure out a way of training fruit flies so they could then go about looking for the genes easily that were involved in learning. Now, of course, there's no guarantee that the learning in fruit flies is going to be similar to the learning in higher animals, which is what people are really interested in. But there's a long history in all of biology, and certainly in all of neurobiology, that all of the basic processes seem to be very conserved. They're the same in us as in jellyfish, for the most part. And so it was a good bet that if they could find genes that were involved in learning in fruit flies, those same genes would be involved, maybe not in exactly the same way, but they would be involved in learning in mammals.
What's the lifespan of fruit flies?
I actually don't know, but I think probably just a few days or weeks. Anybody know what lifespan of fruit fly is? Any biologists in here?
I mean, it's just things like a week. Like I didn't even know that.
Oh yeah, they live that long. Oh yeah,
Okay.
oh yeah, yeah. They live. I don't know. I'm not sure exactly what it is. It's, I would guess weeks. I don't know. In any case, so it took a long time to figure out a way of training fruit flies. But this was done actually not by this guy named Tim Cully, but by his mentor. The techniques that they came up with was this. You have a lot of little training epochs, little short 300-second long training epochs. And during each one, what you do is you present two different odors, odor A and odor B. And when one of the odors is present, you give the animal a shock, electric shock. Then, after you train the animal in this way, you put him, essentially, you actually don't do it on one animal, you typically do it on a whole bunch of them at the same time. You put them in effectively a little T-maze with odor A on one side and odor B on the other side and see which way they go. And of course, if they've learned, what they'll do is they'll go away from the odor that they were shocked, that was present when they were shocked. That works. But what Cully did is to now compare spaced and mass training. In one group of flies, he gave them 15-minute rests between each of these little epochs and the next. In the other group of flies, he gave 3-minute rests. He then, so he's got two groups of flies. He tests both groups of flies under 1 day, and they all know it. Short-term memory in fruit flies is a little bit more complicated than in us, actually. There are several different forms of short-term memory of various durations, but anyway, less than a day. They all knew it. But if you tested them 7 days later, the guys that had gotten the space training remembered it, but the guys that had gotten the mass training didn't. So he's got this basic phenomenon in fruit flies that Ebbinghaus discovered on himself. Now, having done that, Tully is a geneticist, so he thinks in terms of what genes and chemicals and proteins and all that stuff are doing. And so he came up with a hypothesis about what might be going on. He knew about it was in his lab, not his lab, but it was done at Cold Spring Harbor on the East Coast, and that is where Krebs and 1 and Krebs 2 were discovered. So he was very familiar with all this stuff. His idea was shown here on the left. Well, I'll just say it up here and then I'll spell it out for you. Tully's idea was maybe he hypothesized. He says maybe each training trial causes both good and bad Krebs to be tagged. We already know the good Krebs strategy. Bad Krebs, he hypothesized the bad Krebs was tagged too, and that once it was tagged, it would prevent the good Krebs from working. And he also proposed there was slightly more bad Krebs in each cell than good Krebs, but that the tag on the bad Krebs is lost faster than the tag on the good Krebs after a learning trial. So imagine you have a training trial right there, one of those little epochs of training. Okay, what would happen according to his hypothesis is that both the good and the bad Krebs, both the bad Krebs shown in red and the good Krebs shown in black, would get tagged. But there's a little bit more of this guy than this guy. But what he's also hypothesized is that the tag on the bad Krebs, so initially the bad Krebs prevents the good Krebs from acting because there's more of it tagged than there is of the good Krebs. But the tag on the bad Krebs tapers off a little faster. And so as soon as the bad Krebs gets down low enough, so there's more good Krebs active or tagged than there is bad Krebs, we can get some transcription and ultimately make some protein and ultimately do a little bit of memory stabilization. So that's his idea. And he says, if that's the way it were, if that were the way it was, then if you gave mass trials, you'd never get into this green region. You give a trial, and before you got to the green region, you give another trial, and you always keep the bad guy more tagged than the good guy. But if you give space trials, you would after each trial, you would get a little bit of protein synthesis that occur. This is his idea. But then he made a prediction. He says, okay, this guy's a geneticist. He says, I know, or I can figure out, how to make flies that had either extra good Krebs or extra bad Krebs. How to breed flies like that using molecular biology techniques and so on and so forth. And he said, my prediction from this idea would be that if I make flies that have extra good Krebs, then there won't be a spacing massing effect anymore. They'll just learn no matter whether it's spaced or massed, because it will always be the case that there will be the green area right after the training trial. If, on the other hand, I make extra bad Krebs, they'll never learn, no matter what I do, no matter how much I mass it, no matter how many massed or spaced trials I get. So that's his prediction. So he did the experiment. Okay, this is just what I was on the previous slide, so these are his predictions. Now, we have three kinds of flies. Normal flies, flies with extra good Krebs promoter, extra good Krebs, and flies with extra bad Krebs. Extra good Krebs, extra bad Krebs. Let's look at the normal flies first. And we're going to look at 10 space trials. We're looking at 7 days. We're going to test them 7 days after training, and we're going to train them with 10 space trials, 1 trial, 2 mass trials, or 10 mass trials. The normal flies, if we train them with 10 space trials, remember perfectly well after 7 days. But if they only got 1 trial, they don't remember it. Or if they got a bunch of mass trials, they don't remember it. So that's consistent with what we already do. Let's look at these guys, the blue guys first. The guys that have the extra bad Krebs, these ones over here, it turns out that no matter how we train them, they just don't remember anything after 7 days. They all learn it, but they don't remember it after 7 days. The learning isn't shown on the slide. It's just the 7-day test they show. But the one that really clinches it is the ones with the extra good Krebs, because it turns out they do learn it with 10 space trials, as you might expect, but they actually learn it in just 1 trial. They have photographic memory. Tully was very proud of himself when he did this. It's a very, to make that prediction and actually have it work is quite amazing, and it did. And then similarly, for them, mass trials are just fine, of course. It's just 1 trial is fine, and mass trials don't do any harm. So that's a very neat prediction. Well, that's well and good. But as a neurobiologist, and I'm sure this is true with Tully, he would have said, okay, I think this is probably true for mammalian and human learning as well. But, of course, that does have to be proved. Given the history of the similarities between the way the nervous system works, the basic processes, the basic mechanisms within the nervous system, the cellular mechanisms within the nervous system work, it would be expected that something that worked on fruit flies would work on mammals in general and people in particular. But nevertheless, something like this you'd want to show. And so the experiment's not been done on humans. Kind of, it hasn't. But it has been done on rats. So now we're going back to fear conditioning in rats, which we know is mediated by the amygdala, by changing synapses in the amygdala. And the details of this don't matter. I don't pay much attention to the left side. But the bottom line is what they wanted to do is they wanted to elevate good Krebs selectively in the amygdala. And the way they did it is they made a virus that would basically inject good Krebs into the amygdala. Put the virus in the amygdala, and it made good Krebs. And the details of that don't matter. It just tells how but molecular biology techniques for doing it, which we won't go into. The important point, however, is that when they did this, they have 2 groups of flies, or 2 groups of rats. One group is normal. One group has the extra good Krebs being active in the amygdala because they put this virus in that makes good Krebs. So the elevated amount of good Krebs in the cells there. And what they find is they train the animal with mass training, and look 2 days later, and with rats, 2 days after mass training, they would have, they really forget rapidly. And indeed, the normal animals had mostly forgotten, but the guys with the extra good Krebs did not forget. So that is exactly the same kind of result they got in the fruit flies. So everybody believed from this, and that this also works in mammals. Now, as I said, the scientists who did this were basically doing it for disinterested reasons. They just wanted to understand how all this stuff worked. But once they saw this, they saw dollar signs in their eyes. Because they thought, oh boy, if I can find a drug that I can give people by mouth that will do something similar, that will raise their good Krebs or lower their bad Krebs, I'm going to have people that can learn, do long-term learning better and faster. Now, that's a little, there's some large jumps of reasoning there, but you can, it's not a totally unreasonable thought. And so these experiments that I just told you about led to a number of startup companies. As far as I know, none of them have been successful. But this is the origin of the whole article that you may not read until after the midterm, but which you read in the SMART fill. The smartness refers to improving memory. Excuse me. And this kind of stuff does often lead, or can lead, to very practical results. This one, as far as I know, has not yet. So there are, this is all kind of interesting, and it's relevant to your everyday studying. But I want to make a couple of other kinds of points about it. The first one is that the role of good Krebs in memory and a bad Krebs in the spacing massing effect emerged from work on cyclic AMP and controlled genetic transcription by cyclic AMP that had absolutely nothing to do with learning. All the stuff about Krebs and so on was discovered before anybody thought of actually doing experiments on it in relationship to learning. It was done because they were studying other kinds of cells. And it took it was decades and decades of research that led to those discoveries on these other kinds of cells, which were important for other kinds of reasons, or which people were interested in for other kinds of reasons. A moral to that story is that going after something directly, if you focus too much, it's not necessarily a good idea. You often are going to need things from all over the damn place in order to solve a particular problem. And if you just do focused research, you're going to very likely not to be nearly as successful. I mean, you might be lucky occasionally, but progress is usually going to come out of putting together information gotten God knows where, put together in new and interesting ways. But that information will be gotten for a lot of different reasons. And you can't just say, I want to solve this problem and put the information together. Lots of times putting together will occur in kind of an accidental way as a result of having a bunch of knowledge in your head gained from a bunch of different places. And so focused research is great. If you want to solve a problem, you may well want to focus research on it. But it's not always going to be the fastest, best way to progress. I say that because there is a huge tendency in, at least in neuroscience, and I think probably in all science now, but certainly in neuroscience, now, for granting agencies and so on, the federal government, to want to give money to projects that are focused, that have what they call a translational aspect to them, that you can see what value, what practical value it would be if the research were successful. And that's fine. I think there should be research like, exposed like that. We should pay people to do that kind of research. But if that's the way you go, if all the research you fund is of that kind, if that was all the kind of research you funded, it may well be that these discoveries would not have been made, and the startup companies, which admittedly haven't so far worked, never would have started. On the other hand, having gotten this far and having discovered good Krebs and bad Krebs and the relationship to long-term memory, that's the point in time when maybe you do want to have translational research. You do want companies, you do want research directed towards applying it, because now the problem is that the basic knowledge is there, but people haven't figured out how to actually make it work in a practical way. So this kind of research is a good idea, but it's a bad idea to try to make too much of the research that goes on in the world be of this kind. So that's one point. The other point is that because a lot of the same learning mechanisms and principles seem to operate in animals as disparate as snails. We haven't talked about snails, I guess. I used to in these lectures, but fruit flies, mice, and us. The same principles seem to operate. And that's true of basic cellular, all kinds of basic cellular mechanisms. They're very, very general. They're conserved throughout evolution. But often it is the case, as with the use of fruit flies, which were advantageous because you could do genetic experiments on them. It's often the case that you can find out the most about something by studying an animal that's just suited for studying. And so it may well be that it's probably the case that the discoveries on good and bad Krebs still wouldn't have been made if we were just working on mammals. They were made, it turned out to be right when we studied mammals, but we only thought to do the experiments because of what had been done previously on the fruit flies. And so lots of times, scientists will do experiments on funny animals or funny kinds of experiments. You say, why the hell? Who gave them a grant to do that? And the fact of the matter is, the people that think that don't understand very much about the nature of evolution and biology, and the fact that a lot of these processes are highly conserved, and that it makes sense to study things where it's easy to study them, not necessarily where you want to apply them. So that's the one of the morals of this story. Okay, I did finish. We will have a review session, in quotes review session on Friday. Come prepared, try to get some study done, come prepared with questions. Again, I haven't told you this, but let me just say I have kind of told you. For this block of, for this half of the course, with the exception of Dawkins, most of the questions will come from lecture. Not all, but most. But for the evolution lectures, probably be about even between Dawkins and the lectures. Okay. Yeah.